As you all know, we're here today to talk about a shortcut to reliable diagnostic, control, and assay antibodies. My name is Anthony. I'm the Director of Business Development at Rapid Novor. Got here my colleague and friend, Richard Park from Evitria, the Global Head of Client Relations. And in case you like to see a different version of us, here we are. I'll be making the slide available at the end as well, info, so you can always grab that from us if you have any further questions after the webinar. As a quick agenda, just to kind of set ourselves up here, we'll be going through a couple of introductions as we are, discussing some commercial workflows. We'll be going into the technologies that each of our respective companies offer to address some of the most common issues and challenges that we run into with our clients in those commercial workflows. We'll be examining a few case studies and of course having some time at the end for Q&A, as well as letting you guys know where we'll be in the next several weeks here as the spring conference season continues to roll on. Oops. So I prepared this slide. I really try to give us a high-level overview or conceptualization of many of the common challenges and workflows that a lot of people that we work with run into. So, of course, with many people in the antibody industry, I should say, your workflows can look something similar to this, right? So you have like your ideation, where you're discussing your ideas, you're thinking about a new target, you're thinking about new assay or developing a new antibody. You decide it's a good idea. You move forward with this assay development, this procuring of the antibody of interest. That antibody could be commercially available. You could be the ones making that antibody. You could be doing custom work. You're looking for a custom-made antibody. But that's where the R&D is happening. You've selected a good antibody. You've chosen it because you know that it works well. It is robust. It works to a high level fidelity in your assay of interest. And then you want to make more of it. So do the upscale manufacturing before, of course, you put that antibody of interest in some kind of like test, whether that be a commercially available kit for a diagnostic test of some sort, or whether that just be a tool antibody in support of a different program, perhaps a therapeutic antibody program. With this kind of framework in mind, we have found that we have been able to address a number of issues, in particular, issues with respect to reproducibility and quality. A common case that we'll encounter is the loss of access to a hybridoma antibody, for example. It could be the case that over many decades of a hybridoma living in a freezer, that the hybridoma may have experienced some genetic drift. There could be a loss of performance due to other contaminations within that hybridoma cell line. Nonetheless, access to the same monoclonal antibody that you had maybe some time ago is no longer present. So there are ways in which, like Richard and I are able to offer some of our technologies in which we can mitigate these kinds of issues. We have the ability to also help with issues of commercially sourcing an antibody of interest. Perhaps maybe it's not a hybridoma issue necessarily, but for other reasons, the antibody of interest that you've been relying on for many years in your assay is no longer accessible. Sometimes antibodies become discontinued. Sometimes they become prohibitive to access for one reason or another. And in these kinds of instances, we're going to show you guys a handful of ways in which we can actually mitigate these issues or still actually procure access to these antibodies of interest. And lastly, sometimes people just want to, do R&D on antibodies. Sometimes having a good proof of concept molecule can really serve us before we engage in a lot of custom antibody development work. So perhaps, you're looking to develop a bispecific antibody. You just need a good antibody that binds to your one arm. You don't want to make a custom like CD3 antibody, for example. Procuring a good antibody and engineering it in-house for research purposes only is also something that some of our clients have been taking advantage of. And this is ultimately in the name of leading to greater reproducibility, greater quality, and improving access to the antibodies of interest. So this is like our conceptual framework that we'd like to kind of present to you here. And of course, like I said, we'll be going through those challenges as we roll through the webinar. So a bit of background on us, at Rapid Nova, we've been around in the industry for, we just turned 8 actually. We've got many thousands of products that we've been able to complete with many of the top pharmaceutical companies and biotechs worldwide, many major academic institutions and publicly funded institutions as well. We are more recently known for our ability to sequence polyclonal antibodies as well, so not just monoclonals. We're quite proud of that achievement, no lie. we're quite excited to be able to work with these complex mixtures in pursuit of not only therapeutic antibodies, but also good reagents. Of course, like some people just have good, golden goats, golden rabbits, what have yous. And of course, we've been able to work with a lot of these types of polyclonal antibodies and derive the most abundant and usually the most functionally relevant monoclonal antibodies from those polyclonal mixtures. We've got hundreds of clients worldwide, and our team is growing. I've been around since almost the beginning. I was like the first hire at Rapid Nova, and we've steadily grown now to well over 70 members. We've got over 20,000 square feet of lab space that we've built out here in Kitchener, Ontario, just outside of Toronto. That's a bit about us. Richard?
Yeah, absolutely. Short history on Evitria. So hi, everyone. Great to meet everyone. So Evitria's been around for over a decade now. We're on our 13th, going on to 14th year of operations, and in the biotech world, in the contract research organization space, or the contract development and manufacturing space that we both play in. 13 years is quite a long time, so we're really excited to be able to hit that mark. One thing that is unique to us is that from day one, all of our production has focused on transient protein expression in CHO. CHO is kind of the workhorse cell line that is able to produce material for you reproducibly, reliably, and a lot of our work in protein expression has been on the recombinant antibody production side of things. So on that note, we've had over 120,000 successful antibody production runs. If you look at some of our competitors, you'll find that we are kind of above and beyond many times over the production run capacity. And even this year, as we continue to expand, we're in the middle of quadrupling our capacity just because of the continued growth and the amount of business that we continue to see. We are based out of Europe. I'm personally based out of the Boston area. However, our headquarters are based in Zurich, Switzerland. And that's where all of our materials and our reagents are shipped out from after they've been custom produced for you. Obviously, we serve the diagnostic market all the way from initial idea development all the way to commercialization. We can support in any way, any aspect of that timeline. But beyond that, we're well suited to support anyone who needs antibodies. So whether that is at the smaller scale or at the larger scale, or if you're looking to commercialize and you need that additional documentation, we are well suited to support all of those needs. And very excited to kind of walk through this journey with you to kind of to demonstrate those shortcuts and make sure that you have the best reagents available to you as you look to commercialize your diagnostic products. Pass back to you.
Great. And as a kind of running theme, we've also incorporated these tags for the general workflow that we operate within, so that you guys can be aware of like kind of where each one of our technologies plugs into this framework as well, with respect to antibody sequencing, monoclonal and polyclonal antibody development, batch testing, and QC work. And lastly, custom catalog development. I think the latter sections are really more relevant to the evitria side of things, whereas like us, Rapid, are going to be focusing on this antibody sequencing workflow for the most part. We also do some, I guess, some developability and characterization work as well, which they'll be able to get to in the case studies. But in short, yeah, our sequencing workflow is well established, quite robust, and I'll be getting into the technology in just a moment. It doesn't take too long, just a few weeks to do a monoclonal antibody and just a couple of months to do a polyclonal antibody. Okay, so what are we doing? Well, at Rapid Nova, like I said, we've been able to build our technology off of this mass spectrometry-based approach. So we are sequencing using bottom-up mass spectrometry and doing de novo antibody and protein sequencing. So the way it works is we initially take in a sample of interest. Here we always run a gel first. We always want to see how the antibody excuse me, how it looks on a gel, because we want to ensure that what we think is in the sample is truly in the sample. So we'll do a quick gel, take a look at it, and then we'll do our sample prep chemistry. So we're going to be using multiple different enzymes to cleave the antibody of interest into its constituent peptides. I'm going to move a little bit fast here, but I have some slides that'll make this very clear. Once we do our sample prep chemistry, we'll be injecting those samples onto the LC-MS system, and this system will output many spectra. Those spectra will be interpreted by our algorithm into sequences. And then once we have the many smaller peptide sequences, we can actually assemble them into a full-length contiguous heavy and light chain. In the case of a monoclonal antibody, it's not too challenging to imagine which ones pair together. I'll get into the polyclonal side in a moment. But once we have those prospective chain sequences, we put them into the report for you to interpret and read, and perhaps contact evitria to express. So a little bit more detail, so it's not so fast. Sample preparation, as I said, we started with different enzymes, and you'll be seeing some data in a moment as well. The different colors here actually denote different sample enzyme chemistries. So we have, a handful of different enzymes that we use, but each color in our data is specific to the enzyme that was used. The reason is because they can cleave at different sites depending on the enzyme of interest. So we have these peptides that we've made. We don't know the sequence yet, but they're different enzyme chemistries. We put them on the mass spec. The mass spec will, as I said, generate some spectra. Our algorithms will then take those spectra and output peptide sequences. And again, because we knew we've labeled them with the correct chemistries, we know which ones belong to where, and our algorithm is actually going to be able to take these, truly thousands of different peptide sequences and rearrange them into one full-length contiguous protein sequence, as you see in our image here. So that's for the monoclonal antibody workflow. And for the polyclonal antibody workflow, it's quite similar, except we do have a few other aspects to which we are trying to purify the sample a bit more. So in the polyclonal antibody sequencing workflow, we start off with an immunized animal of interest. We have the luxury of working with many different animals, as I'll show you in the next slide. But typically what we end up doing is we take this we take the serum from the animal of interest and we purify it against, firstly, we usually run a protein A or G purification to reduce the polyclonal antibody population to just IgGs. And then we antigen affinity purify against your target of interest. We've been able to successfully work with a number of different antigens now. Of course, peptides are quite common. Many soluble proteins, in particular some GPCRs, which are amenable to a soluble kind of format. And we've been able to work with some larger targets as well. So we're happy to say that the technology has been quite successful and robust so far. But it really hinges initially on the purification of the antibodies against your target of interest in the assay or in the functional way in which you expect to see them, right? So this is a really key component, I think, of working with us in this kind of workflow, is ensuring that the antibodies that are in the tube are the ones you want. I have no doubt. I have absolute faith in our ability to sequence the antibodies inside of a tube. It is always about ensuring that the antibodies you want are the ones that are inside of that tube. So that's always, I'm a stickler about that. I just want to make sure that we're always on the same page whenever we discuss this. So once we have these purified polyclonal antibody pool, very similar to what I was mentioning above, the antibodies will be put through our sample preparation chemistries. We will put them on the LC-MS. We'll generate spectra, and we will generate a list of prospective heavy and light chains. We've also developed a proprietary method for pairing the prospective heavy and light chains together, so that we have at the end a list of paired cognate heavy and light chains, so that we can express the full-length IgG, again with perhaps someone like evitria. And then you can use those recombinant monoclonal antibodies to whatever needs you have. Optionally, we can also work with the NGS data that we would derive from a PBMC extract. So we've done this now in a number of species as well. It is truly an optional step, although I would say that it would help to increase the breadth of the antibody space in which we can explore. Although in many instances, the B cells are not necessarily accessible, that's not a problem. We've definitely been able to work with a lot of projects and do truly de novo antibody sequencing directly from the serum of the immunized animal, or perhaps an older bleed that's been around for a while. One of the only other key conditions that the serum come from a single donor, one animal. It can be taken from multiple time points, but truly just one animal. So one rabbit, one goat, one alpaca, what have you. Okie doke. With that in mind, as I said earlier, we've been able to work with close to a zoo's worth of antibodies from multiple different donors. We've been able to successfully work with, human antibodies. We've done some mouse work, a lot of rabbits, done a couple of canine projects. Alpacas are quite newer, but a much more one of our more interesting cases as well. Goats, I have some data to show you a little bit later on. Sheep as well. So we've been able to really demonstrate that we're not restricted to one particular biology. We can really explore, a great variety of different antibodies that exist in a great variety of the animals that are used for traditional antibody development, not only in livestock, but also in some unique applications like companion animals, as well as obviously one of the bigger cases, which is the human case, which we'll be able to talk about a bit more, not during this webinar, but in the coming months, I believe. And of course, if you're unable to conduct a fresh immunization, or if you've just got this golden goat, golden rabbit, what have you, perhaps you've got a catalog of antibodies that you already acquired. These are perhaps reasonable antibodies for us to be exploring for sequencing as well. And maybe you've just got a bunch of antibodies that are sitting in your freezer that you've been saving for a really long time. Indeed, in today's world, it's possible for us to sequence those antibodies. We've recovered a lot of antibodies, actually, monoclonal antibodies from like the freezer, in terms of like a recovery type of experiment where you're hanging on to the last little bit of this monoclonal antibody you like to work with. We've been able to recover and sequence many of those types of antibodies, but as I said as well, a lot of people just have a lot of polyclonals that they developed over the many years, and they've been saving them for quite a long time, again, for similar points of interest. It can be sometimes quite challenging to recapitulate the results of an immunization program, right? So in these situations, of course, Rapinova has the means now to access the functional antibodies that are present in those samples. And once we have those sequences, it becomes quite a bit easier to do a lot of the engineering and expression types of work that I'll let Richard comment on now. So let me just switch things over.
Excellent. So thanks, Anthony. I think that's a great overview, and I love where you left off, because when you start having the sequence reports that Anthony, ideally, you send many things to Anthony to get sequenced, and then that results in many sequences for you to comb through. What we found is that many of our clients, when it comes to truly identifying, okay, this commercial product or this hybridoma-based antibody that I've been working with, I want to make sure that I'm getting the exact same type of activity. I want to make sure that it's behaving the same way, but maybe that's not the only thing that you want to do with it. Maybe you want to re-engineer it. So the most requested modifications that we get once Anthony's group shares sequences with us is, okay, do we want to change the species out so that we can have different species-specific binding? Or do we want to figure out ways that we can switch out the subtype without modulating the activity in order to increase titers, for example? Because sometimes a hybridoma might produce an antibody at very low expressions or low titers, and then the recombinant version might produce better, or vice versa. And then the third is, are there ways to modulate the activity? And so again, that goes into sometimes subtype switching or species switching, depending on whether this is a kind of a primary or a secondary antibody or a capture or detect antibody. So all of those engineering options are capable once we have the sequences. We have a library of roughly 600 different constant domains with many different types of mutations. All of the kind of hot and popular ones that are most commonly used are also within our library. So we can very easily switch things out as long as we have the appropriate variable domains that are provided by Anthony's group. And so knowing the sequences from remAb and repAb allow for any number of customizations to specifically align with what you eventually want to get out of this diagnostic goal that you have in mind. So beyond engineering, in terms of how we support the development of it. So you have the sequences, now you have to go back to the bench and figure out how you're going to make sure that what we produce works on your end. So for that initial development phase, what we would suggest is producing between 1 to 10 milligrams per construct of the monoclonal or the polyclonal to make sure that it goes through all of your internal validation needs, and we would support the production of that. Once those are indeed validated, you've identified which ones you exactly want to move forward with, then we would move on to providing 1 or 2 additional milligrams for batch testing and QC development in order to make sure that the batch that we produce day 1 is the exact same material that we produce on day 100 or year 4 down the road. Because on our end, we're not looking to be a partner for just a quickie. We want to be there with you in the long term. And so because of our very industrialized approach to production, we can always make sure that when we produce material for you during this initial validation phase and development phase, that's the exact same material that you're going to be getting 2 years, 3 years, 4 years down the line. And so because of the standardized industrialized approach, again, that we've had and developed over the past 13 years, this is something that we can provide. And then on the end, when you're looking to actually develop this further and then commercialize, there's no upper limit to the amount of material that we can produce. So depending on kind of how widespread your diagnostic test might be, sometimes we have individuals who say on the commercial side, we only need maybe 5 to 10 to 100 milligrams of material. We also have clients who say, hey, by 5 weeks from now, we need 2.5 grams of material. So depending on the scale of diagnostic commercialization you're looking for, we can support any project with no upper limit. Logistically, what does it look like? So like I mentioned, all of our material is produced in Zurich, Switzerland, and our timelines start from the moment that you approve a project all the way to shipment of the purified material. So the initial production will always land between 5 to 8 weeks for the molecular biology work to get all of the DNA prepped and ready, and including the actual scale-up, expression, purification, and analytics, and aliquotation and shipment. Then all of that material gets to you. You go through the validation, and then you let us know, hey, variants 1 through 10, we'd like to exclude from future work. We've validated that they do not work, but we would like variants 11 through 15 reproduced at a higher scale. And when you go through reproductions with us, because we keep the plasmids in-house for anything that has been previously developed, we can provide not only the 0 lead time guarantee, but turnaround to shipment somewhere between 2 to 3.5 weeks, depending on the scale you're looking at. In addition, I skipped over the middle bullet point there. All of our projects have a 0 lead time guarantee. So when we send you a proposal, you now have that in your inbox to say, hey, Richard, or hey, Evitria, we'd like to initiate this project. The very next business day in Europe is when those projects and that timeline will start counting down. There aren't any queues, there aren't any wait times, and there's no worry because, again, of how industrialized our approach is. I think I mentioned this before as well, but over the next year, we're also going to be quadrupling our capacity. So this 0 lead time guarantee is even further highlighted. In terms of further mAb and pAb development, there is the internal QC that's provided with the development phase. And so that would provide the protein purity, the protein quantification, the protein concentration, protein endotoxin measurement, as well as the OD and extinction coefficient. This is all information that comes standard with every production and every production run that you do with us. I'll go through an example of kind of what kinds of things we've seen in terms of purity, quantity, et cetera, in a case study at the end. But this is the initial QC specs that are provided with every construct. Further development, a lot of our clients need these antibodies, but they need further work on them as well. And a lot of that comes from custom protein labeling. So you've had the antibody developed, you've had it produced by us, and you also want to add it with some fluorophores or small molecules or additional proteins. Those are all things within our wheelhouse as well. So we have an additional 1 to 2 business weeks for the post-purification labeling with a new certificate of analysis provided with the labeling itself. And again, there's no upper limit of labeling within this. So clients who need 1 milligram of a standard antibody that they use very rarely, biotinylated, happy to do that. If you need regular bi-monthly productions of an antibody with PE labels, then that's also easily done. All of this is done and all of this is coordinated in concert with what you have in mind. And at the end, when the custom catalog is developed, all of this filters in and gets consolidated so that it becomes a seamless experience once we're past this development and co-development phase. So that leads into the batch testing and further QC development. So let's say you're working with the FDA or you're working with another regulatory body that's saying, hey, I need to see a little bit more documentation around the fact that batch 1 produced year 1 will be the same as batch 500 produced 5 years from now. And so this is relatively need dependent and can include anything from activity and affinity-based testing, back-to-back batch production and testing, additional LC-MS work, as well as peptide mapping and more. But again, very need dependent, and not all of our commercial partners currently even need this level. So it's really dependent on what your ultimate needs are and how you're approaching your market. And then lastly, like I mentioned before, the custom catalog development. It's jointly developed, so it's truly a one-on-one exercise. For my clients who I work with on these custom catalogs, I sit down with your procurement team, your vendor management team, and we work together to understand, okay, how do you like to have material procured? What is your ordering process? And we tailor the catalog to make sure that when you order something and you provide us a catalog number, that is in line with a milligram amount, already includes the additional QCs that you may or may not desire, and all of the expectations is boiled down into a catalog number so that the ultimate users know that if they have a catalog number that they give to their procurement team, they will get that material within a couple of weeks exactly how they expect it to behave. And so all of these catalog numbers capture the previous developmental work and ensures that the material day 1 is the same as material day 100. So with that, I will quickly pass it on to Anthony for additional case studies.
Sweet. Thank you, Richard. And I think we're back. Yes, we are. Very good. Thank you. Yeah, so we have a few different case studies that we've been able to work on in the past year or so, especially, well, several years, but like really the past year we've been working on this one in particular, which was our alpaca case study, which shows that we were not only able to sequence the most abundant monoclonal antibodies from the serum of this immunized alpaca, but we were also able to really quite robustly characterize it as well with some of our newer technologies that we've been using, in particular SPR. So this project went through our typical repab antibody sequencing workflow. This was an alpaca that was immunized with a rabbit polyclonal antigen. And with that, we were able to purify the antibodies. Most of them were nanobodies, although I will also point out that because we were actually sequencing the functionally expressed antibodies, and because we've actually developed some of our own techniques for enriching for the entire polyclonal antibody and fractionating it, we can actually access not just the heavy chain only antibodies, but also full length IgGs that exist within the camelid serum. There are a few different isotypes that may be of interest. I'm just kind of pointing that out. But of course, most people come to us with a camelid project looking for nanobodies. So we offer something akin to this. So as I was saying, so we take the serum, we purified it against our targets of interest. We did our digestions, our separations, and we put them on the LCM SMS, and we generated some prospective sequences, which we were able to characterize, as I said earlier, using SPR. So in this case, we had our analytes, our alpaca mAbs as controls, as well as these R numbers. Numbers are referred to the antibodies of interest that we were able to actually sequence directly from the serum of this alpaca. So in the case of the ligand was some control antibodies against some rabbit mAbs. P17 was an internally developed antibody as well from a rabbit, and a CD3 epsilon was your CD3 ligand as well. This is a carboxyl sensor, and it's a typical situation that we find when we're using SPR. So we really found that of the 10 antibodies that we were able to sequence, 6 were of greater interest to us, and so we ran those antibodies here on the SPR that we use in house. So we use the Nicoya Open SPR for our workflows as well, just so everyone knows. So we found that these antibodies we generated were in some cases quite high affinity. Of course, we have excuse me, our binding graphs here showing that R9 and R7 look pretty good. R4 looks also pretty good, but in particular, some of them are cross-reactive. Obviously, R9 in particular is cross-reactive to both P17 mAb as well as the CD3 epsilon mAb. We were also able to validate these antibodies for their binding activity orthogonally using ELISA. So we found that the ELISA results did in fact confirm the results that we observed on the SPR as well. So we're quite happy to see that these antibodies perform really quite well in both assays and demonstrate that functional activity in this cross-assay format. And we have a bit of a Venn diagram here showing you, the activity of these antibodies with in how they engage with their antigens of interest. Lastly, we have this iso affinity plot showing that a couple of these antibodies, in particular R4 is an extremely high affinity antibody that's picomolar binding and subnanomolar binding, or sorry, nanomolar binding as well. These other antibodies here. So, this is kind of some of the characterization workflow that I was mentioning earlier that I wanted to make sure that we were able to highlight was that we've been able to onboard not only this kind of like affinity binding assay workflow, but we've also been able to validate the epitopes using HDX mass spectrometry as well. So we're trying to really address a lot of the early stage R&D needs at Rapid Nova. So we're not just doing the sequencing and generating an antibody of interest. We're also able to robustly characterize these antibodies using things like, well, obviously your ELISAs, SPR for affinity, kinetics, epitope binding, binning in this slide as we see, but also binding to the specific epitope of interest and validating that at the amino acid level using hydrogen deuterium exchange mass spectrometry. But of course here on this slide we're showing you that we're actually able to do some binning experiments where we saturate the sensor with one of the antibodies of interest, and in the example here that I'm showing, we're saturating the sensor with the R9 antibody, and then we ran so that the R9 antibody is binding to your antigen of interest, and then we flow over the other antibodies that we had. So R7, R4, R1, in order to demonstrate that in a competitive situation, these antibodies do or don't bind to the antigen in the presence of R9. And on the bottom here, we can see that R9 shows that it outcompetes R4 and R1, we see relatively low levels of activity with these antibodies as they compete with R9. But R7, as we see in the sensogram, demonstrates that there's actually R7 outcompeting R9 in this interaction. So we are able to band these antibodies together and demonstrate that they probably target a very similar epitope, if not the same epitope, in this way, which we can then use as an indicator of, well, if you really want to know the antibody epitope, sorry, the epitope of interest, of course, we can have a much more high fidelity and sensitive interrogation of that using HDX MS as well. Great. And the other case study that I wanted to quickly go over was our goat study. So we were quite happy with this goat study because, as I mentioned earlier, sometimes you have a golden animal. This was one of those cases where the goat generated an extremely powerful antibody response against this target of interest. In this case, it was a peptide. But the peptide was specific to this antigen, your AIF1, and this this antigen was challenging to just generate an antibody response against. So we were able to work with this. This is actually from a partner of ours. They had injected their goat some time ago, many years ago actually. It had been repeatedly immunizing this goat with that peptide of interest in order to generate a fairly mature antibody response. I think it's really important to understand the context here as I show you in the next slide. But this is a fairly matured antibody response after repeated immunizations on this long-lived animal, and we put it through our workflow as you've seen previously. So here's some of the data, as I mentioned earlier. So just so we're on the same page with respect to what we're looking at, this top line here is noting our position in the antibody. So this is your amino acid position from D and terminus. So we're starting with the green is indicating your variable regions. Your CDRs are in orange, and to the right and ongoing in blue would be your constant region. Below that, you have your sequence, which we're obviously obfuscating in this instance. And then you have the lines. Each individual line, as I mentioned earlier, refers to a peptide that was observed on the mass spec that we sequenced using our algorithms. And the different colors denote different sample enzyme chemistries that we use to prepare the antibodies prior to injection on the LC-MS. Essentially, the more lines, the better. The more coverage, the better. Although, we can get into the weeds of this about like how specific each individual line is, or how valid every line is. But of course, the consensus is that more lines is better. So in this case, we're able to demonstrate that the data was quite deep for this project. Like we had very strong reads throughout this antibody sequence. And actually, we can't even show you all of the depth because we can only fit so many lines on one screen. But suffice to say, the data looked quite good. We were quite happy to see that the heavy chains from this antibody had fairly robust coverage. And from there, we were able to successfully express these monoclonal antibodies from the polyclonal mixture and show that they faithfully recapitulate the binding activity of the original polyclonal antibody mixture on a couple of different ELISAs, actually. So really, we found that there were two monoclonals within the polyclonal mixture, and that's why I was bringing up this fact about the goat being long-lived and having many immunizations and having a matured antibody response. When we do a lot of these polyclonal sequencing projects, we don't know what we're getting into necessarily with respect to the number of antibodies that we expect to see. In this case, very small antigen, many immunizations over a long period of time. I think we're closer to the more conservative end of things. So we actually only really observed two monoclonals within the polyclonal mixture, but there was still a polyclonal background. On the flip side, there could be dozens, if not hundreds of antibodies that are present, but really at high concentrations, it's usually no more than a few dozen antibodies that we're able to observe on the mass spec that are we believe, conferring that functional activity that you're, really seeking to recapitulate. So, but luckily for us, these two monoclonals, like I said, worked really well on the ELISA, and they were able to be used in a handful of other assays as well. So we found that they worked well not only by ELISA, but also by flow cytometry, by immunofluorescence, and we're able to really demonstrate the cross-functional activity of these monoclonal antibodies, and really, again, faithfully recapitulating the original polyclonal antibody that was so valuable. Like, I think that we're really proud of these kinds of projects because, of course, they tried to make that polyclonal again, but couldn't. It's like sometimes you just get really like quite lucky with these immunizations. They generate an antibody of interest, and they can be really challenging to recapitulate or expensive. I mean, you could obviously immunize 15 goats and see what happens, but that can be pretty prohibitive, like to a lot of people, in particular for due to cost, let alone the like actual scientific risk that's inherent to such a project. So working with the functional antibodies that work, that have been able to be demonstrated to be efficacious in your application of interest, is usually like a much more de-risked kind of starting point. And so for that reason, we're pretty keen to help people in those kinds of situations, really recover those antibodies and meaningfully move forward your project of interest. So that's it for those case studies that I got, and then I'll hand things over to Richard.
Yep. So the single case study that I wanted to discuss should be on everyone's screen now, and it's what it looks like for kind of an overall project. What does a long-term relationship look like? So this is a client who came to us and said, hey, I have a bunch of products that I have in terms of reagent kits that my company sells, and we also sell capital lab equipment to go along with it. But all of the kind of individual antibodies that we've been putting into these reagent kits are getting quite expensive to source from a commercial vendor. And so we're thinking of putting together the investment in order to make sure that all of these reagent kits can be produced more cost effectively and recombinantly, so that one, we're not at the whims of a commercial provider, and two, we have much more control over the ownership of this reagent kit. And so they came to us and said, hey, we'd like to work with you. So quickly after that, we started receiving the sequences. And so over the course of our relationship to date, we've produced over 100 unique constructs for this client. And that is across 161 production runs. And each sequence that they provided to us, because sometimes these were based on a hybridoma, or sometimes these were based on the commercial antibody itself that they had sequenced. Kind of an inside secret is that not all of these materials that say that they are pure when sourced commercially are actually pure. And so we found ourselves on the production side needing to produce between 1 to 4 variants per sequenced antibody. And so that's why over 100 production runs led to 161 production runs led to the generation of just 100 unique constructs. And so from this, there was an average purity of roughly 96.4%. On Evitria's end, when it comes to producing, because we have a standardized approach, the purity or the amount of aggregation that you might see once a material is produced is highly, highly dependent on the sequence that is provided. And so when we see lower expressing material, or when we see kind of very close to no production of material, even though we go through the production process, that typically means that that variant might not be the right one. And so there are some times when we produce a variant very easily identifiably, that is not the one to go for. So in the end, we are now at a point where with this client, we have 45 constructs that we've put into a final custom catalog. We have guaranteed the ability to deliver the material to them in increments of 10, 50, 100, and 200 milligrams. We're working with other clients going through the same process, and those catalogs are actually looking to be delivered in the increments of 1, 5, 10, and 20 milligrams. So these increments are very, very dependent on what exactly you're looking for. And then the catalog pricing will remain valid for 24 months. So that gives you this ability to make sure that, okay, I'm investing now and I can kind of plan out financially and logistically how much it's going to cost me to make sure that I can get this off the ground while maintaining a fixed cost for actual reagent sourcing. And depending on the amount of order that's depending on the volume of the order that this client actually provides to us on a monthly basis, additional free analytics are provided, because at a certain point, it behooves us to make sure that if we're happy, you're happy. Sorry, I think I definitely just said that inversely. So if you're happy, we're happy. And so we just want to make sure that overall we're establishing a good relationship, because like you see in the title of the case study, we do want to keep this very, very long term. So with that, I'll actually pass it back to Anthony to present the final couple of slides.
Great. Thank you very much, Richard. And should be all set now. So that's that's really about it from us. I guess, in conclusion, hopefully we've been able to make the case that, despite some challenges that may occur with respect to access to monoclonal and polyclonal antibodies for your applications of interest, there are ways in which we can recover a sequence and produce in large quantities, as well as engineer those antibodies of interest, so that you can really take advantage of good antibodies that you've been working with for some time in whatever application of interest you might have. So with that in mind, we hope to see you soon. We'll be on the road. We'll both be at PEGS in a couple of, well, like next week.
Next week.
Yeah. Obviously pretty close for you guys, for you, Richard. Working out of Boston, but I'll be coming down from Canada as well. Our whole team will actually, and so we'll be at booth 1206 for Rapid, and you guys will at Evitria will be at 304. I think you guys will be pretty easy to find. I hope that we're easier to find this year than last year. But we're both in the convention center, so I'm sure you guys will be able to find us if you're looking. And then beyond that, I and my colleague Dylan will also be going back to Boston and going to the Bio International Convention as well in the first week of June. So
Great.
That's where you guys will be able to find us. In case you've forgotten, we're also available by email. You can reach us email on the emails above, but of course you'll be getting a follow-up email after this webinar as well, with access to the recording, and of course you'll be able to initiate a dialogue with us there should you desire. So I can leave that information there for our slides and open it up. If there are any questions, please feel free to submit your questions here. If there are no questions in the next couple of minutes, I'll shut things down. We can always chat afterwards as well. So let me know. But yeah, I think that we're open to take your questions at this time. You can pop your questions into the chat. So if you're viewing, it should be accessible through the bottom toolbar, and then there's a chat function or Q&A, whichever you'd rather. I'm watching both.
Anthony, I have a question for you, if anyone online is still interested in hearing a couple of things. What would you say in your process people are most surprised to learn about? Like, what makes them go, oh, like I had no idea this would have been part of the process?
The answer that jumps out to me right away is how little antibody you want is how little the fraction of the antibodies you want represent the total fraction of antibodies in an immunized sample. You know, that so for example, like, you know, people ask like, you know, how much do I have to bleed this animal in order to get enough antibodies for you guys to sequence? And so we like to start off with like 1 milligram of antigen affinity purified IgGs to run the project. And so that come from 20 mils of a bleed from like a rabbit or something like that. But it could be more in some instances, it could be less than others. It's quite an interesting thing, you know, like we start off, we will obviously do a protein protein A purification in the instance of like a rabbit. So we have just the IgG fraction, and then let's say you get 100 mgs from that initial 20 mil bleed. That 100 mgs, you know, it could be just 1% of that fraction that's antigen specific, so that we get that 1 mg from the entire 100 mg fraction. It could be even less. It could be like 0.1%, you know, and it could be 3% of the antibodies that are specific to your target of interest. So that was kind of like one of the more interesting things that we've been able to, you know, somewhat figure out here. I don't want to like generalize and say that we've got this thing nailed down, right? But like, because it's biology and, you know, things are always different. But I would say that's probably one of the more interesting and surprising things is that number, that antigen specificity, can really vary a lot depending on the antigen, the animal, the age, the circumstances of the immunization, the bleeds, so much that can run into that, you know, because it's such a dynamic system. So yeah, that's probably one of the more interesting things that I would say. Yeah.
Makes sense.
How about yourself? Like, what's clearly caught in a lot of minds?
I'd say in the context of this conversation, it is definitely the idea that a commercially sourced antibody or an antibody that's commercially sourced that comes from a hybridoma is not as pure as the available COAs say that they are. So sometimes the commercial constructs will cost hundreds of dollars for micrograms of material and supposedly have greater than 95% purity. And then you see the actual reports that come out from your sequencing results that say, hey, there are multiple heavy chains present in this pure sample, or there are multiple light chains available in this pure sample. I think the worst case that I've seen is having to produce like 6 variants for a single antibody that was sequenced, because there were, I believe, like 3 light chains and 2 heavy chains that were all present. And honestly, when we're needing to produce that much material, there's a benefit that you can easily see, okay, this matchup will definitely not produce any material. There's very low titers, there's very poor purity. So those are things that can easily be knocked off. And then you have some where both variants end up producing adequately. And so you have to invest the additional time into doing the internal validation on those constructs. And it'll always be an investment of time and resources. But I think in the end, what our clients walk away with is kind of the knowledge that they have the right sequence and that sequence is theirs. Because on our end, once you share the sequences with us on the production end, we're purely a service, pay per service company. So we retain no retain no IP, and anything that we produce is fully yours, including the COAs. So we try to make sure that that's always abundantly clear. But it does require sometimes significant feedback from us, because we have so much experience in antibody production that, hey, this matchup is likely not going to produce anything. Would you like us to go ahead with it? Or we know that if you want to switch these variable domains into a mouse IgG2A backbone with those constant domains, you're going to see a huge increase in titers. That's feedback that we often have to provide, because so many people are coming to us having kind of only used commercial products throughout their diagnostic journey, and that feedback is necessary for them to kind of feel comfortable in pursuing production projects with recombinant.
For sure. Yeah. I can imagine if you've never really gone through the process of making a recombinant, that there's a lot to it. Which is why I'm glad you guys are around.
Yeah. I haven't seen any questions come in. Have you?
Nope. Me neither. So with that in mind, I don't think we're gonna get any. So
All right.
that's fine. If there are any other questions, please, everyone, you'll be receiving that email soon, and we'll be able to follow up with you quite quickly. But without dragging on too much more, I think that we'll leave it here. So
Sounds good.
yeah, I think that's it. I will wrap things up here. Just want to make sure we say goodbye.
Anthony, it was great talking to you and hanging out with you again as always, and see you next week.
Sounds good, Richard. Same. Yeah, I'll see you guys soon.
All right. Bye,
All right.
everyone.
Bye-bye.